I'm Kurt Marsden. I'm an assistant professor in the Department of Biological Sciences at NC State. The goal of our work is to really establish a basic understanding of how the nervous system is put together and how neurons wire together and form synapses with one another, and how those circuits that are formed are regulated through the activity of genes or environmental factors such as exposure to toxins. And we are currently doing that with three basic approaches. We're using a forward genetic approach that I started as a postdoc, where we did a random mutagenesis that created mutations throughout the genome, and then we screened through thousands and hundreds of thousands of baby fish larvae to look for mutant lines that had a specific defect in a behavior called the startle response. And so we have a set of mutants that came from that screen that are very easily startled and perform a startle response following a very low intensity stimulus, which is akin to part of what happens in some folks that have autism, also in anxiety disorders and schizophrenia. Many of those folks have a heightened startle response. And so one of the motivations for that project is to try and understand some of the molecular regulators of how the startle response is controlled. And then another approach that we take is to do a reverse genetics approach, which is to target specifically a single gene that we know is affiliated or associated with a disease called CHARGE syndrome. This is a gene called CHD7, and in human cases, this results in a number of issues, including heart issues, gut issues, reproductive issues, as well as a whole slew of behavioral and psychiatric issues. And so we know very little about that side of the disease. And so we set out to try and understand how some of those behavioral and psychiatric issues arise in CHARGE syndrome by creating a zebrafish model of that disease. And then finally, to look at environmental factors that influence behavior, we're collaborating with Michael Behrman's lab, and our graduate student, Rubia Martin. She's been doing a project looking at how exposure to cyanotoxins, which are produced in algal blooms, how those may be linked to ALS, or amyotrophic lateral sclerosis. And so we found some really exciting results that show that exposure to a mixture of two cyanotoxins can potently increase the startle response in baby zebrafish. And we have done some proteomics work to try and identify the molecular pathways that drive those changes, and we're really excited to follow up on some of those studies. I think our work is really important and really exciting in that we are identifying some fundamental mechanisms about how sensory information is processed and is translated into behavior. And for example, we study the startle response, which is associated with things like autism and anxiety disorders. And we know relatively little about how the threshold for triggering a response is established. And so by understanding in a fish what those basic mechanisms are that wire that circuit together and control how that circuit functions, we can hope to have provide some understanding of what might part of what might be going on in some of those conditions. And then for that goes for ALS as well. We really know very little about most of the causes of ALS, and there's strong association with environmental factors. But really teasing apart what those mechanisms are and what those factors might be is, I think, really important in terms of trying to understand and help to prevent and to diagnose and to treat those conditions. I'm Kurt Marsden, and this is how I think and do in the Department of Biological Sciences at NC State.
When you were little, what did you want to be when you grew up?
I think I wanted to be a baseball player, like every 8-year-old boy in the Midwest.
Who were your favorite players?
Ozzie Smith. I was a Cardinals fan, so
Okay.
he's the greatest.